Hi, everyone. So I'm Noelle. I'm a graduate student at MIT and Woods Hole Oceanographic Institution in chemical oceanography.
And I'm Saunders, a NASA postdoctoral fellow at Woods Hole and MIT.
So we have basically an applications talk for you today. So we're going to show you a little bit about how we're applying scientific Python to our questions about the ocean, and hopefully share with you a little bit of knowledge about the ocean in general through the process. So put your oceanography hat on for this talk. So we'll start with some background, and then we'll jump into the scientific Python-based applications that we've been building to help ourselves and our collaborators to understand this data that we're generating. So it's probably no surprise to this audience, but the ocean is a very dynamic place for microbial life in the ocean, in the world. So what we're showing you here is the distribution of different functional groups of phytoplankton in the ocean over the course of a few years. This is based on an MIT Darwin model. So you can see that the distribution of these cells in the ocean is changing over the course of time. And what we as microbial oceanographers are interested in is understanding, first, what are the distributions of these cells over the course of time, and then what drives the distribution of these cells. And we really think that the chemistry of the ocean is a major player in sort of understanding how these cells are distributed in the ocean. And then by contrast, how those microbes impact the chemistry of the Earth is really important for us to understand the general Earth system and the health of the Earth system. Next. So the way that we study these systems is using metaproteomics. So that's like a loaded term. It's a very big word, but basically it's the study of proteins in the ocean. So you've probably heard of genomics, which is the study of genes, and proteomics is the study of proteins. So proteins are beautiful molecular machines that actually perform many of the chemical transformations that biogeochemists are interested in. So if you think about, this is one of my favorite proteins, nitrogenase. So it converts dinitrogen, which is like basically the atmospheric version of nitrogen, to ammonium, and it breaks a triple end-to-end bond, which is almost impossible to do. So it's a really beautiful molecular machine, and it basically acts as a fertilizer for the ocean by providing nitrogen that is biologically available to organisms in the system. So we measure these proteins in the ocean, and we try to figure out how that connects to ocean chemistry and the general Earth system. So we're going to be showing you data today from a specific research expedition called the ProteOMZ expedition. So we studied proteins in a region of the ocean called the oxygen minimum zone. So these regions are really cool because they're basically places where the system is nearly anoxic. And they can provide us with a picture of the past ocean, since we think the ancient ocean was largely anoxic. But they also provide us a picture with the future ocean, since we think these oxygen minimum zones are growing as a result of climate change. So you can see, this is the cruise party here. That's me. I came right off this cruise, actually, and just a few weeks later came to my first SciPy conference ever, so that was pretty fun. So this map is showing you the distribution of oxygen in the ocean at 300 meters depth, and warm colors mean more oxygen and cool colors mean less oxygen. And overlaid in these little yellow stars are the cruise or the expedition stations, so the places that we sampled in the ocean. And you can see that we sort of sampled a gradient of oxygen concentrations in the ocean. And the goal was to understand the biology and chemistry of these unique regions. The way we sample the ocean is by deploying in situ pumps that pump hundreds of liters of seawater through tiny filters. And these filters collect cells, basically, that are living in the water mass. And then we use tandem mass spectrometry-based methods to analyze the proteins in these cells. So we're not going to discuss these methods in detail, but Jackie or I are happy to talk to you about them if you're interested. The data then goes through a series of bioinformatics and statistical pipelines. And then finally, what we generate are basically spreadsheets of 60,000 or more microbial proteins and basically their relative abundance at different locations and different depths in the ocean. And this creates a major bottleneck for us and our collaborators who are trying to then make sense of what this means about ocean health. So this particular data set is about 10 stations, actually 11 stations, with 8 to 16 depths sampled per station. If you measure 60,000 proteins in each of those samples, you end up with 6 million individual data points. So how does an oceanographer make sense of this? In addition, this comes along with thousands of what we call metadata, which are distributions of physical or chemical features in the ocean. And our goal is to connect these proteins to this metadata. And so that created a major challenge for us. I'll note that this data analysis bottleneck is just beginning for the ocean metaproteomics community. So I'm showing you data from this transect right here. But all these red dots represent locations that we have samples either analyzed or in the freezer ready to be analyzed. So we're going to just be generating more and more of this data moving forward. So it's really a unique challenge for us as a field and a really cool opportunity. So Jackie's going to tell you more about the chemistry of this region, then we're going to jump into some actual scientific Python work.
So I'm just going to go over general oceanographic concepts, as well as Noelle said, talk about some of the really unique chemical features of the oxygen deficient zones. So here we have a depth profile, which is kind of a bread and butter data visualization for oceanographers, as well as geologists and soil ecologists. On the y-axis here, we have depth, so this would be the surface of the ocean going down to the depths. And on the x-axis, we have density, or really you could plot any feature that you're interested in, so salinity or temperature or nutrients. So here we're specifically looking at the density profile. You'll notice that it's this kind of vertical line here, and this is the surface of the water column, and so the density is uniformly distributed because this is a really well-mixed region of the water column. Then you'll notice this dramatic change over a very short period of depths. So dramatic change in density over a short period of area, and this is called a cline. And so it really separates out these two regions of the water column. So you have a mixed surface layer, this kind of density barrier, and then a slower mixing, deeper water column. So now I'm going to just look at this kind of section, but over a distance. And so here we once again have the surface of the ocean moving to depth. And I've got that dramatic cline represented by this change in color. So within this well-mixed surface community, we have life. We have all of these lovely photosynthetic microorganisms that are taking in that sunlight, fixing carbon dioxide into organic material, and using photosynthesis. So this region is very oxygen rich because you have that active photosynthesis releasing oxygen. You also have that part of the water column interacting with the atmosphere. But because of all of this photosynthetic activity, these waters are also very nutrient poor because all of those nutrients are being taken up by those primary producers. So if we look at the bottom part of the water column, this water column is very nutrient rich because it's interacting with the depths, and as there's many geologists in the room, that's going to be the primary source of nutrients, is going to be interaction with the seafloor. There's also life here, even though it's dark. And I just want to show this very generic equation for respiration. So go back to your high school biology class. I'm sure you all vaguely remember oxygen is used to oxidize organic material to harvest energy, releasing carbon dioxide and water as a byproduct. So how is the life respiring down in this region? I've told you it's oxygen poor in these oxygen deficient zones. Well, I'm going to steal an analogy from Chip Breyer at University of Texas, Rio Grande. You can think of the microbes as basically little computers going through if-then statements for metabolic activity. So if there's oxygen present, they will use the oxygen. So as it is the most energetically favorable electron acceptor, releasing 820 millivolts in this redox potential. So in these oxygen-poor waters, the microbes are using nitrogen compounds, as that is the next most favorable electron acceptor, using nitrate and reducing it to more reduced nitrogen compounds. So there's a reason I'm telling you this, and that's because we have built a data visualization platform to look at these various oceanographic features in this particular environment. Okay. So here we're looking at a platform that we constructed to view some of these oceanographic features, and not just one at a time, but looking at this whole panel at one time, so you can really get some nice oceanographic conceptualization of a transect. So here's that transect that Noelle described earlier, and Here we're looking at all the different features. I want to hone in on some of the stations within the oxygen deficient zone. And now I'm able to interactively work with this data thanks to Bokeh and explore, okay, here I have my oxygen concentrations, and you can see very high in the surface. You see that dramatic climb where it drops down, and at depth we have these lower oxygen values. What's really nice is that we're able to just highlight these points that we're interested in these stations, but still have oceanographic context for the rest of the transect by changing the alpha value of the points that are not selected. So as I talked about, nitrogen, so these oxidized nitrogen compounds are the next most favorable electron acceptors in this region. So if we go in and look at the nitrate and nitrite concentrations in this oxygen deficient region, we can see they're very low in the surface because nitrogen is also a nutrient, so they're being drawn down by all of those photosynthetic organisms. And then it increases at depth and then is drawn down again because it's being used for respiration purposes. What's really nice is we also have this table, thanks to Bokeh, that highlights the data that we're currently looking at. And we can also sort within this table. So if I'm, say, interested in, let's see, the oxygen concentrations, I can sort here as well and look for the values where I have the highest oxygen concentration or the lowest.
So one of the nice things about this in particular is that it's an HTML file, so it's one of these Bokeh standalone documents if you're familiar with the platform. So we've been sending this to collaborators who are not Python literate, never installed Python ever, and they can use it. And so that's what's really nice about this particular platform, really thanks to the sort of the Bokeh standalone documents, and I can't get out. So we also built a platform to visualize the metaproteomics data. This is not a Bokeh standalone document. It is actually built with Bouquet Server. So what that allows us to do is basically take actions that occur in the browser and sort of connect them back to the full power of Python, so we're not limited necessarily to modules that are already in the Bokeh package. We can basically make our own scripts up and then send it back to be visualized in the browser. So again, we have a map based on GMAP, basically the GMAP function in Bokeh, of all the stations that are present. And you'll see that we have all these little widgets which allow you to select on data. So this is a Bokeh Server document, so when you make a selection in the widget, it sort of sends it back and then comes back to the browser. So we'll select a single station. And then this plot here is really the bread and butter of what we're trying to visualize out of this data set. So again, it's the depth starting at the surface and going down. And then the x-axis is the abundance of proteins. And so each protein is plotted as a line, so you can see the abundance of that protein. So I'm going to highlight the most abundant protein that we identified basically in this data set, which is a nitrate oxidoreductase subunit alpha, which we were really excited to hear this word. Basically, it means nitrogen respiration is occurring, and you can see that it's very highly abundant in this region, and it's also only really present at deeper depths where the oxygen is basically very low. So it's really sort of a proof of concept showing that this data is showing us real oceanographic patterns. And what's beautiful about it is that when we select a single line, for example, in this vertical distribution, it sort of populates all of these downstream graphs. So this graph is showing you protein abundance as a function of some chemical or hydrographic data. So we can select, for example, nitrate concentrations, and we can see that it sort of already was on nitrate, but we can see that the distribution of this protein is positively correlated with nitrate concentrations. And if I select oxygen, you can see that it's negatively correlated with oxygen concentrations as a whole. And so that makes sense, since we think these organisms basically can't live when there's a lot of oxygen present in the water column. And so this allows us to basically rapidly probe these sort of biological and chemical connections. So as before, we were basically doing this in Excel and selecting columns and making plots, and it was a disaster. And now we have this platform, which is a lot easier to use. We can also select like individual taxa of microbes, so groups of of microbes. So I just selected Prochlorococcus, which are the most abundant photosynthetic cells on Earth. And you can see they're only present in the surface waters. So the proteins of these organisms are only available above 200 meters, which makes sense. That's great. The data is good. If I select the nitrifying bacteria, so organisms that use nitrogen to fuel respiration, you can see that they're not present in the surface and only present at deep waters. So with just a few clicks, we can sort of rapidly identify these layers of biology and chemistry in the ocean. And we can do this in a way that can allow us and our collaborators to sort of identify hypotheses and quickly test them in this sort of browser-based system, and then they can go probe them deeper later. One thing that you may have noticed is this percentile slider, where it says be patient below 90%. So right now we're only actually displaying the top 5% most abundant proteins in the data set that you've selected, and that's related to performance issues. I don't even want to move this slider right now because it'll be sad. But basically, we really pushed the performance of these plotting platforms. So we tried some other methods. We used data shader, for example, which is one way that sort of you can get around these massive oversampling data sets. So we have an example of that in our proceedings paper, but we didn't have time to talk about it today. But yeah, it's really interesting. It's been fun to really push these packages. I'm going to go back to my slideshow. So we learned a lot as scientists applying scientific Python to our work in general. So one of the benefits and one of the reasons we started with using Bokeh in particular is because it makes beautiful plots. You can see they're all very nice. They look cool in a browser. And a lot of these were meant initially for sharing as like outreach events with children or with donors or someone who's interested in the work we're doing. They're fully interactive, which is really nice. So when you click on one plot, you populate another. And then those HTML outputs are really great. And of course, it's Python-based and open source, which actually isn't necessarily trivial in the proteomics field. It's really nice to have open source platforms. But we ran into some challenges related to sustainability and stability. So one of the risks you take when you work with a version 0 package is that you're going to have changes to it. So our original platform had like a Bokeh donut chart, which no longer exists. And so we quickly had to learn to version control and work with virtual environments and be careful about these things. And that was really important for us to sort of learn as scientists. Also, the Bokeh interactivity relies on all of the data being in a single column data source, which is their data model, which allows for these plots to be connected. So it basically enforces a two-dimensional data structure on our data, which isn't really two-dimensional. So it was really difficult to figure out how to best get that data into the column data source. So we actually brought, Jackie helped a lot with that, and we brought Joe Futrell on, who's another co-author on this work. He's in the room, to help us figure that out, basically. And so, but once we worked through the documentation, it really did help to get it all in the single column data source. And then, of course, I alluded to this before, but we really are pushing the limits of Bokeh that is run on my laptop, for example. So we can only display the top 5% of the data, but we really want to see those low abundance proteins since they're often the most interesting. So there's work to be done there for sure. We have a lot of exciting projects moving forward. So I'm showing you like a little video of me playing around with this data sharing repository and visualization tool called the Ocean Protein Portal. And you'll see that we built a lot of the Bokeh charts. We actually have some HoloViews based charts and Matplotlib charts into this platform that we're now sharing with our collaborators and colleagues in the oceanographic fields. So the idea is to make this metaproteomics data about the ocean publicly available, so anyone who wants to go look at the distribution of a protein because they're interested in it or they need it for their work can easily access and visualize this data. So we learned a lot about the visualization process by building this platform, and now we're sort of learning how to plug it in to an actual data sharing repository website. We have stickers for this project if you want a sticker. We would encourage you to go and check it out and test it. And if you find a bug or something, let us know. It's definitely in beta testing right now. But we're excited to share it with the larger oceanographic community moving forward. So finally, we'd like to acknowledge our co-author, Joe, who really helped us to understand what we were doing and helped us to figure out the right framework to get this project going. And our advisor, Max Saito, who actually did most of the coding for the hydrographic notebook that we showed you earlier. We actually have some of the Ocean Protein Portal team here with us as well. Nick and David are in the audience, so they are helping us to build the Ocean Protein Portal. Word on the street is that they're also looking for more help with some of these projects, so talk to Nick and Dave if you're interested. And then, of course, all of the people who helped us to acquire the samples and generate the data, both in the Saito lab and outside of it. And then, of course, also the contributors to our favorite scientific Python tools. We can't do this work without you, and it's really exciting to see how excited, I guess, you guys are about our science and how helpful that your tools have been to our scientific work. I attended this conference 2 years ago on a scholarship. I'd only been coding for 3 months, and now 2 years later, I'm here and sharing some of this work with you. So thanks for being a welcoming community as well. Thanks.
I think I learned something today. Thank you very much.
Awesome talk and great work. I was wondering if you guys could talk more about the sustainability side of things. I think that, as you know, one of the challenges of building software in an academic setting is you guys are all going to move on in a couple of years. So have you built a plan so this will not all kind of fall apart when you leave, or how are you thinking about for the long term?
Yeah, so in relationship to these platforms in particular, they're all sort of living right now in our own individual GitHub accounts, which sort of puts the onus on the individual to maintain the platform. The Ocean Protein Portal website is more structured. It actually has NSF funding through EarthCube, which is sort of a general data sharing and repository-based structure. So we have people on that end. Actually, the original funding we got for that was basically a prototype. So what we're showing you now is the prototype. And then, yeah, sustainability is a major question moving forward. I'm sure also Nick and Dave can probably talk about the scope creep. We're really good at scope creep. And so it's really important for us to work with computer scientists who can help us to make sure that we're staying within the realm of what can actually be done and kept and maintained, basically. But, yeah.
Hi. This looks very cool. I'm curious about how easy, in theory, it would be to, people go on these cruises and get other sorts of data too. How straightforward would it be to display other people's samples instead of the samples that you guys have been focused on in particular?
Yeah, exactly. We should have discussed that a little bit more. So basically it's fed from a CSV file, which is a common output for metaproteomics data. It's fed from a common output that many metaproteomics scientists use that basically comes out of the bioinformatics and statistics software. So the idea is that they could just download their CSV and then put it in. And then the hydrographic data is similar. It's basically an Excel file, which is what most of our collaborators are used to working with right now. So
I'd like to make a comment on that. So the hydrographic, that set of code is really nice and easy and kind of just the same way you would look at it in an Excel file. You can plug it in, it'll work. However, the metaproteomics viewer, as Noelle talked about, the column data source, we did have to create a weird way to manipulate the data to get it into a two-dimensional framework. However, in the proceedings paper, we outline exactly how we built that, kind of kludged all of it together to make it two-dimensional. So yeah, you can find more information there.
We have plenty of time still left for questions.
This looks like a really great tool. I'm really excited to try and put my data into it. Could you go back to the slide that has the GitHub address?
Absolutely, yeah. So we actually wrote it up as a proceedings paper too, so it's all in the SciPy GitHub account.
Okay,
yeah.
Thank you.
Yeah, that helps.
Great talk, and those are some really beautiful visualizations. I was wondering if you could maybe talk a little bit about your experience working with the Bouquet Server and maybe give some tips or things to look out for?
Absolutely. Yes. So basically understanding it was difficult to begin with. That's why we brought Joe on board initially. Like figuring out the documentation was difficult for us to sort of work through. So the general, the way I understand it is that basically the column data source can act as sort of an intermediary between Bokeh and like the background JavaScript that basically Bokeh does for you. And so I don't think we actually have any JavaScript actually in the code. If we do, it's just a few lines. But so it's basically, I don't know if I'm really answering your question very well. Maybe Joe can explain better, yeah.
Hey.
So without Bouquet Server, your script has to write callbacks in JavaScript. And you can do it by writing them in Python and they get translated into JavaScript. But the problem is they don't have access to the backend data then, and they can't change the column data source that everything is based on. So if you need that kind of callback, you have to use Bouquet Server. And that requires a slightly different setup. And then you have to run the server on a port and talk to it, and it gives you the HTML, and the callbacks go back, and magic stuff happens, and data moves from Bouquet Server into the column data source, which is then accessed by JavaScript. That's a little tricky to orchestrate, and it's a little tricky to migrate from an app that doesn't use Bouquet Server to one that does. But you still get a lot of heavy lifting done by Bokeh itself in writing the callbacks and linking them to the different JavaScript widgets. It all looks like a big Python app either way. So we found that extremely useful, but we had to navigate all of the ways that you connect everything to everything else in Bokeh, which required poring over documentation and trying things and seeing if they crashed. And then also, I mean, Jackie and Noelle have alluded to this, but getting multidimensional data into Bokeh is basically black magic. And we had to do the black magic ourselves.
I will say that, so the Bouquet Server framework that Joe helped us to build is now on that GitHub repo, and it might be useful, I hope it would be useful for others just to see the framework and how we organized basically the callbacks and everything.
We've got time for one or two more questions before we go to the next talk.
Are you also collecting metagenomic and transcriptomic data? And if so, are you visualizing it in a similar way?
Yes, so we collect metagenomic data especially because our metaproteomics analyses really rely on the metagenomics. We also sometimes do collect metatranscriptomics, and I think the idea would be that basically any large omics data that you have abundance for many different biological molecules, you should be able to use this visualization platform to see.
Any more questions? I have a quick one. So you've got a very nice visualization tool for data discovery here. Is there an ability, or do you plan to have an ability to annotate data? I picked this is cool, you should check this out, and actually have that data discovery history stored for sharing with other researchers.
I see. We haven't really discussed it, but that would be a really excellent idea, I think. Yeah, it'd be a good way to help people sort of crowdsource the discovery process.
It seems like such a rich data set that there's a lot of, unlike my boring numerical data, this is much more long tail data that human eyes and brains need to sort through. Yeah. Any more questions? Okay, can we thank the speakers again?